says he belongs to life science background. Can you let me know what is your educational degree as well? Are you from a microbiology or a biology background? Let me know in the chat. And it would be great if the rest of you could introduce yourself in the chat box. All right, so Vikas says he has done his master's in medical laboratory science. So I'm curious to know, what made you interested in the field of bioinformatics? Do you have a specific research interest in mind? So for those participants who have just joined, we are just introducing yourself in the chat box. You can let me know, are you from a biology background or from a computer science background in the chat box? I'm monitoring that. We will be starting with the webinar in just a few more minutes. All right, we have Festus with us. I hope I'm pronouncing your name correct. Otherwise, you can let me know. So Festus is from a computer science background. That's pretty cool. Can you let me know what made you interested in the field of bioinformatics? Do you have a specific research interest in mind? All right, so Festus says he has paper in bioinformatics and is at master's level, but still learning and wish to do PhD in bioinformatics. That's pretty interesting. Do you have a specific research interest within bioinformatics? We do have a couple of students from computer science background who have been doing projects on machine learning. Do you have any such interest? Let me know in the chat. All right, so meanwhile, the participants can introduce yourself in the chat box. I'm going to start with today's webinar. Hello everyone, a very warm greeting to one and all. My name is Sri Gauri Krishnakumar, and I would like to welcome you all for today's webinar on Introduction to Bioinformatics Multi-Omics. So, this online program is designed for everyone, including students who don't have a background in bioinformatics and biologists, to learn about NGS data analysis processing sections such as genomics, transcriptomics, and metagenomics. And as a result, the participants will become proficient in analysis and interpretation of various types of omics data using bioinformatics, including commonly used statistical and machine learning techniques. In today's webinar, we will first learn to sign up on the Omics Logic Learn portal and learn how to navigate the various courses and example projects that you will be completing as a part of the program. Followed by this, we will have a hands-on session by the program mentor where you will learn to run bioinformatic pipelines for various sections such as genomics, transcriptomics, and metagenomics. Finally, we will also cover details about the program registration process and program cost. So before we begin with today's webinar, let me also take a brief moment to introduce you to the team behind the program. We are a US-based bioinformatics company who's working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. And as a part of the program, students will have access to the Omics Logic training that has been completed by over 24,000 participants from 187 countries in over 300 workshops. And due to this fast growth, our team is working with local and regional coordinators that are helping refine local program logistics and adapting them to the needs of students and researchers around the world. And this training has been completed by participants in 6 different specialization tracks that includes oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data, and comprehensive training on omics data analysis. Students will also have access to the Omics Logic resources, but how does it help advance bioinformatics teaching? Omics Logic is a portal for practical and theoretical learning of bioinformatics. It takes a combination of training modules, data analysis tools, curated project datasets, and interactive sessions with mentors to give the student a clear path up the Bloom's taxonomy pyramid. And no matter what the user's initial experience with bioinformatics is, Omics Logic is a useful tool for gaining a baseline knowledge of the theory behind bioinformatics, analysis of large datasets, and introduction to basic coding languages like R and Python, or even beginning a data analysis project. And this also comes with an access to the Omics Logic courses, which includes courses on bioinformatics, precision medicine, multi-omics that consist of genomics, transcriptomics, metagenomics, and epigenomics, data science that makes use of bioinformatics for R and Python, as well as cheminformatics, and example projects of different fields which you could learn from and replicate for your research work. Now, as a part of the program, students will also be given access to the T-Bioinfo platform for bioinformatic processing and analysis of data. The platform includes demo pipelines as well as data management and analysis cloud infrastructure to run the bioinformatic pipelines, and different stages of analysis are performed in different sections on this multi-omics platform. More on this will be covered during this webinar. So now, to view the various courses, example projects, student projects, you need to first sign up on the Omics Logic Learn portal. Before that, let me walk you through a brief demo on what a completed profile on the portal looks like. So once you sign up on the portal, these are the features that you need to update on your account. That is, a completed profile will have a profile image, full name with appropriate capitalization, link to your social media handles, and a brief bio about yourself that will consist of your educational background and research interests. And the mentors, under the activity tab, the mentors can view your learning progress. And under the courses tab, you will be able to view the various courses that you are currently completing. Finally, after the completion of your course, the certificates will appear under the certificates tab. So this was all about the Omics Logic Learn portal. So now let us begin by signing up on the portal. For that, I'm sharing the link to the portal in the chat box. So please click on that link and I'll show you how you can sign up on the portal. So for those participants who are creating an account for the first time, you can simply click on the create an account option that you see here. And here you can enter your name, your email ID and password. This is one way to sign up on the portal. Or you can also sign up using any of your social media handles. This could be your Google, Facebook, Apple account, GitHub account, or even Twitter. So you can simply click on that and you will be able to sign up. So I'll pause for a couple of seconds and I'll be monitoring the chat. Once the participants have signed up on the portal, please put a yes in the chat box so that I can show you the various courses and example projects that are there on the portal. And along the way, if you're having any queries or any technical issues that you're facing, you can put that in the chat as well, and we'll be happy to look into it. I'm monitoring the chat. Please click on the link and sign up as I explained. Please put a yes in the chat box so that I know I can proceed. For those participants who already have an account on the portal, you can simply click on login now option that you see here and simply click on sign in. All right, so Vikas says he has signed up. All right, so I'm going to proceed along, but along the way, if you're having any queries, let me know. So since I already have an account on the portal, I'm just going to click on sign in. So once you sign in, you will be able to see the various courses and example projects that are there. So when you scroll below, you can see a search bar that asks for looking for a specific course. So here you can enter your topic of interest. So since most of you are beginners in bioinformatics, you can simply bioinformatics, and that will show you the very introductory courses that makes use of the bioinformatics concept. So let's say you have a very specific research interest, such as machine learning. So you can simply enter the keyword machine learning, and that will show you the various courses and example projects that are there on the. So this is how you can navigate the various courses that are there on the portal. But for now, let's go on to the part where I was discussing about updating your account. For that, you can simply click on the welcome back with your email ID tab that you see here. So once you click on this, this will be displayed in front of you. So here you will be able to see an icon that says profile. Just simply click on that. And here you will be able to edit your profile. So as I explained in my presentation, you can enter your name. Make sure that it has to be in proper capitalization. And you can also enter a brief bio about yourself. So that will be your educational background and research interest. So in my case, it's autoimmune diseases and neuroscience. So I've entered that. And also please make sure that you update a profile image for yourself and also put a link for your social media handles. So in my case, I've linked my LinkedIn account and my org ID. So that was about updating your profile. So this is the activity tab where the program mentor and the community manager will be tracking your learning progress. And once you start taking up any of the courses, it will be displayed under the courses tab. As you can see, I'm currently completing transcriptomics analysis in Python. So that is displayed here. And once you complete a project and upload it on our portal, it will be displayed under the projects tab. So once you enroll for the program, such as the Multi-Omics program, that will be displayed under the programs tab. And finally, after the completion of your coursework and the associated quizzes and assignments, the certificates will appear under the certificates tab. So this was all about navigating through the portal. And you can also take a look at some of the student projects that are there under the projects tab. You can take a look at a wide variety of the projects that students have completed on topics ranging from infectious diseases, oncology, neuroscience, machine learning, and so on. So with that, if the participants have any queries, please let me know in the chat and I'll be happy to help. Has everyone signed up? Because today is going to be a hands-on session. It is important that you sign up so that you can make use of the resources that are there. All right, so if the participants have no further queries, we can start with the next session, that is the hands-on session by the program mentor. So for that, I'd like to invite Dr. Raghavendran Lakshminarayan to start with today's session. Over to you.
Thank you very much, Sri Gauri, and thank you for everybody to chime in about your introduction. And I've been following everything. Very nice. We have a good set of participation who are interested and also who have some introduction to bioinformatics also. I see that Cynthia has raised her hand, and if you have any query, please, yeah, let me know. If not, then we can move ahead. Okay. I think we have a request for sharing request to help with the website link for signing up.
Yes, I'll reshare the link. Okay,
thank you very much, Ojasri, and please signing up. So usually what happens is after you sign up, we will have to distribute your credentials for the server, which I'm going to demonstrate to you today. So we had planned for some participation from you also to work with us while I'm demonstrating you, but due to some technical reasons, that's not going to happen. So what you can do instead is you can get the recording of today's video, and then you can use your credentials and then work with me when in the server and the server's capability, which we are going to discuss today, in your own leisure. Right? Okay. This will also work if you already have your credentials to the Bioinfo server, which we are going to discuss in detail today. So if you are a repeating participant, and if you have already signed up earlier and you have credentials to log into the Bioinfo server that we are going to talk about, please let me know in the chat box, and you can sign in to your Bioinfo server account, and then you can follow along with me. Okay, let me find ways to share my screen and set it up. Let's share everything so that it doesn't hiccup in between. Okay, please confirm that you can see my screen. I'm going to be monitoring the chat while we are discussing. So if you have any questions, please put it up in the chat.
Yes,
can you see my screen? Yeah.
Page.
Okay, excellent. So let's go over the program page to understand what do we have here and how do we navigate between different resources, and what are the general syllabus and course structure, program structure of this specific program. So you can find the schedule, and think you would have already navigated towards the schedule earlier. So I'm not going to go over the schedule and those details. So we always start with an introduction session, right, where we are going to introduce bioinformatics to those who are new to bioinformatics. And this is not only specific to people who are new to bioinformatics, but this is a concise introduction, a concise overall view of bioinformatics and its application in various different areas, ranging from agriculture to treatment response and patient stratification, and etc., right? And pandemic response, etc. Everything that you can think about using high throughput sequencing or next generation sequencing technology to find answers in biology, we will give you an overview on that in this specific course, right? In this specific session. So if you already have some introduction, so you can actually wait for, or you can go through some of the additional resources that we have in this case, like Bytes and Molecules, Introduction to Bioinformatics course, and gain some more familiarity in the area that you already know, and also in some of the areas that you are interested in, but you lack familiarity. And those who are starting afresh in bioinformatics, this is a course that you would definitely want to complete, both the video session, and also the associated resources that you can see from here, because this will be the stepping stone for variety of different terminologies that we are going to discuss, and different ways and different logic that we are going to discuss in the upcoming sessions. So you can straight away access part of the online resources, associated online resources, by clicking onto this link, right? Let's do that. Okay, it's opening in the same page, so I want to go back because I don't want to lose this one. So what I wanted to demonstrate to you is over the course of providing you the resources for each and every session that we have been discussing, that we will be discussing in the lectures and in the videos, we would have handpicked a specific lesson from the course, from the Learn portal for you to refer to. Okay, so in the Learn portal, as Srigauri demonstrated you, we have courses, and each and every courses are separated into different lessons. In most of these cases, we would have referred to specific lesson from this for associated resources that we are discussing in the lecture, so that you can quickly refer to what we have been talking about, and and some of the methods that we are discussing during the session. But throughout the course in which the lesson is actually referred to, the course will contain more information regarding other facets of the same of the same topic, right? Let's not take Introduction to Bioinformatics, where we have referred to the whole lesson in itself. Let's go down, for example. Let's take this associated resource of visualization of 16S metagenomics data and Introduction to Metagenomics, right? So if you go here, this is going to be very specific to what we will be discussing in that particular session, right? So you can, for example, look at how to visualize and analyze metagenomics data, which we will go through today also, right? So this is going to be an extremely specific lesson, and you can quickly refer to what we have discussed and then here. But over the course of other lessons that is present in the metagenomics course, you can get an idea about other facets of the data analysis. Pardon the slowness of the server. For some reason, it's really slow today. Okay, I just wanted to show you or demonstrate you the lesson and course-wise structure, where we will be referring to one of the lesson as resources. If you have time and if you are interested, you are free to check and go through the other lessons in the subsequent and cases, right? That's what I wanted to discuss here. Okay. Wait a second. Give me a minute. Yeah, I think it is taking time for everybody to load as well. So that's okay. So that's what I wanted to show you. The lesson and course structure will be helpful if you are going to invest in more time, for example, in learning one specific omics data analysis and the background behind it while you are going through this program. Okay, let's go ahead. So this program is actually separated into different omics data types that we will be dealing with, and what are the considerations for performing that analysis. What will you learn when you perform such an analysis? And for example, how to perform this analysis in the associated big data server, Bioinfo server, that we will be discussing in detail today. So this is a link for that. You can quickly check this one, load it up and check this one with you. Okay, and let me find ways to share. Okay, so let's go back to the course structure. We'll come back to the server later. So after getting yourself introduced and after getting yourself a good overview of different bioinformatics terminologies and applications, etc., we will start with the father of all omics data analysis, genomics, right? So genomics data analysis, we will discuss about, for example, what are we looking for when we are performing genomics data analysis, right? And how do we define, for example, a variant, which is what we are looking for, and how do we define a mutation, right? And what are the effects of variance, functional effects of variance, and what are the functional consequences of finding a mutation? And when can we annotate a variant, and how and when can we annotate a variant into, for example, some clinically relevance, and with clinical relevance, and with toxicity parameters, etc., associations that we will also discuss. So this is, while discussing this, I will also show you how to perform such an analysis in the associated lessons Bioinfo platform, right? And before even going to the analysis, we will also discuss what are the file formats that we are going to deal with, and which is how the data is stored into different file formats, how the data is actually kind of interchanged between different file formats, which is going to add up to the complexity and also to the knowledge of the knowledge that you can gain from the data. So let's quickly, for example, go into a demonstration of how a genomics data analysis can be performed in the big data server, Bioinfo server. So those who have login credentials to the server, you can log in and then follow along with me, right? So in the genomics, and under the genomics analysis, under the genomics section in the Bioinfo server, so we will find this mutation analysis and the mutation variant and the parallel analysis of variant data. So we have to go through that, but to help you understand how to set up a pipeline of analysis using some preloaded input files and preloaded parameters, we have prepared a lot of demo pipelines, right? So these demo pipelines can be accessed using these drop-down option under each one of these analysis pipeline options that we have here. So wherever you find this drop-down option, there's a demo pipeline inside that you can test. So today we are going to deal with, or we are going to discuss, let's take breast cancer mutations TP53 demo pipeline. So it is addressed as demo, etc. So you click on this. So when you click on this, you will get more or less the same screen that you will get when you click on the analysis pipeline link, right? Except for the fact that because we have preloaded the input file, you will not get the option to load your input files here. So, but other than that, this demo pipelines, or these demo pipelines, will guide you through the process of setting up an analysis for genomics data analysis. The input is going to be FASTQ files for multiple different samples and different sample conditions for which you are interested in studying. So this particular input is to understand variants in the TP53 gene using target sequencing approach. So targeted sequencing approach. So we have already set some of the input parameters and input files here. So we will start with the pipeline, start setting up the pipeline. So you click start, then you will get a short overview of what that particular step is doing to your data. And some, for example, you will get to know further in the next few options. If you have a method that is linked with a citation or that is linked with a research paper, then we give you a small gist of what that method is going to do to your data and what are the inferences that we are going to get and what is the type of output that is going to be passed on to the next analysis algorithm. And if, as I mentioned, when it is published in a when it has a publication and a research paper, then we cite that also. So you can click on the citation and check out some more details about this particular analysis. So notice that after I have pressed start, I have an option that is highlighted with which I can proceed to set up my pipeline, right? So now after setting up the start option, then I have an option to trim my FASTQ files using Trimomatic. So what does this Trimomatic do? How does it trim? And what is the use? And how are we going to incorporate that in the analysis? All that will be discussed in the respective sessions. So now we are going to get an overview how to set up this one and get yourself familiarized with what we are dealing with. Look, we have the citation link, so you can link and then get more information about the scientific background of this particular algorithm. And then after setting up the chemomatic and cleaning up the pre-processing the FASTQ files and the reads, then we are going to set up mapping procedure, right? So mapping is where all of your reads are going to be mapped to that particular place where it can find its match to. So that happens using several algorithms that we will be discussing over the course of this introduction program. But one of the most popular algorithm that is widely used and that is also converted into other forms in the mapping world, right? The mapping algorithm options is a Bowtie-2 algorithm or Bowtie algorithm, right? So we use Bowtie. We can use Bowtie to map our reads onto their respective positions in the reference genome. So that is what we are going to deal with here. Since I did not tell, but it is not. I think it is understood that since this is the data that deals with breast cancer mutations in the TP53 gene, the reference genome is going to be human. Okay. Yes, Saksham, if you have just obtained your login credentials or obtained your registration, then you will have to wait for at least 24 hours to get your account activated in the T-Bioinfo server. So that is why I said it's Bowtie-2. Right? Bowtie-2 is a very popular algorithm. It is used in several other this also, right? So you can click on this one, and yeah, I did. You can click on this and get the respective publication also, right? Okay. So, and after that, we can visualize our mapping of the reads onto their respective places like this. We will navigate such a visualization tool in a while, and you can read through what you can gain from this visualization option in this visualization setup. So we always do that because we want a visual confirmation of how our reads are mapped onto their respective positions. And then the whole idea of this particular genomic pipeline is to identify variants, right? We have, look at this, we have several different variant callers, usually called as, right? And variant calling algorithms that we can get our hands to. When you are going to set up your own analysis, you will have options not only to mutate to, but several of these different algorithms that if you want to obtain or if you want to estimate variance for your sample, you can use that in your pipeline. So now for this one, we are going to use mutate2 and and more information regarding how mutate is going to work and how it is going to find out, etc. So it's a little bit slow today for some reasons. I also checked before, it's a little bit slow. Let's go ahead. And then we have another option, for example, of once you have identified variants, right? Mutate2 will identify variants that are present in your sample compared to the reference genome. It's just going to list out all the variants that you can find in the that you can find in your sample. And once you have found that, once you have this list, right, it's really useful to find out or compare this to publicly available resources that documents the pathogenicity, that documents the annotation in disease-related annotations of these variants, so that we can use that information to further implicate the biological reasoning or biological implications of the variants that we have just identified. So that can be done using the annotation option. So when you select this option, you will get a choice to choose either ClinVar or DBSNP to annotate your variants. So once you have done this, let's click end. So ending the pipeline will take you to the output options that you can download and visualize and navigate around and generally understand what are the outputs and what am I going to do with these outputs? How can I improve my learning from performing such a genomics data analysis by going through the outputs, right? So that's what we give you when you sign up in this program, and when you have your even when you have your demo credentials in the T-Bioinfo server. You have full access to all the demo pipeline, which will again give you access to how we set up these pipelines and how we let you visualize the output in the visualization tools and dashboards and all the pipeline output files, which you can obtain from this list, right? We don't have to download this, but I have already downloaded this, but we don't have to download this. We can use the we can use the interactive visualization tool to gain access to all of these output files. So let's click on this genome visualization option and let it load. So if you check the output files, right, we have a VCF file, list of VCF file that we have identified from Mutex 2. And we even before that, we have mapping stats table, and which is going to give you statistics of the mapping of our FASTQ reads in the FASTQ format to their respective positions in the genome, reference genome. So this will give you an example of how many different samples do we have and what are the files that are used for analysis and what can we learn from this during the visualization option. It's going to open up like this. So in this window, right, or in this option, you will get the mapping statistics table preloaded, right? And some of the important parameters that you would want to confirm and you would want to compare and understand the quality of your different samples that you have given for the analysis is to check the overall alignment rate, right? And how many were paired, right? You see, when these are really high in the orders of 90s, then you have a good data, a good meaning all of the paired end or all of the reads that you find have their partner in the paired end sequencing option or in the paired end sequencing output. And out of those or out of these different parameters, we find 99, more than 99 of them have found their respective positions and have very high alignment rate to the genes or to the position in the reference genome. So this is what we want to check. This gives you also more information about, for example, the quality of the samples right here. So we can find out if suddenly one of your sample has a drop in the alignment rate, then we would want to check what's wrong with the sample and how do we deal with the sample. That's a major question. Do we still use that sample, integrate the sample to our analysis, or do we discard that? That depends upon how many samples do you have and what is wrong with the samples also, that we can also check. So if you have a big screen, that will be very useful, but for simplicity, I've completed going through the mapping statistics, and I'm very happy with the mapping statistics of my input file, quality of the input file. So I'm closing this data or closing this particular option. Okay, then what do we have here? We have several of the tracks, and each one of this is a file, and we can check these boxes to display them here. The order in which we want to display them will help navigating this visualization tool also. Okay. Okay. So, for example, I would like to have my reference genome right on top, and I mean, this is just a preference. For some others, they will want to have their reference genome on the bottom. So I would want to have my reference genome right on top. So it's going to load up the reference genome, and you can zoom into the reference genome. We will do that later, and then we would want to load up the annotation file for the reference genome, which is also which will help you, for example, to navigate through the genome in the range of genes, right? Or else we have to navigate through the genome with the loci and with the positions, genome positions, etc. That's going to be a little bit confusing. So now we have loaded both of these things, and there is there's not much information here, but we will do that. And let's load a couple of samples, or let's load the VCF output here, right? So VCF is also loaded. So why is nothing displayed? That's one of the major questions that we would want to ask, right? If this is the analysis output, why is nothing displayed? So this is where just blind performance or blind analysis will not help you here. right? Remember what I mentioned about setting up this particular data analysis or genomics data analysis. We have set up data analysis for targeted genome sequencing data. So that targeted sequencing data targets only the TP53 gene, but we are in some other position where nothing is being recorded and nothing is being analyzed. So let's go to TP53 gene. When you have your GTF file or annotation file loaded, and then this is the usefulness of the GTF file, you can just type the gene name and we can navigate to that specific position. So once you go there, then you can see that, okay, my VCF file is starting to get populated, and I have their exons and the introns mapped by the GTF file that I have obtained from the reference NCBI reference table, etc., etc. So for simplicity, I'm going to open this up and I'll show what happens. Right? Let's zoom in to this one particular section. Right? We have to go here, yeah, and can select this and zoom to this region. And now you can start to see the sequence. If you zoom in even further, some more. Yes, now you can start to see the sequence, and this has lots of information, but we don't want to have reading frames or amino acid translation in different reading frames now. So I'm going to turn the top, right? Show translation. I turn the top. I want the forward and reverse. So let's have it, and let's rearrange this one a little bit. And okay. Okay, so what do we have here? I don't have any variant in this particular region. So I'm navigating through the variant file to find a variant. Let's zoom out a little bit. Okay, in this particular exon, I don't have any variant at all. That's surprising. That itself is a good information. Okay, we have one here. So let's click on this variant and see what this actually tells us. So this tells us some other characteristics of the variant. And for example, what are the samples in my sample list contains this variant and in what capacity, etc. So these are the information that you will get while navigating this. This is much navigating a map. This is a BAM file and SAM file, right? Visualizing this map, BAM file and SAM file is a very extremely qualitative job. Right? This can this is done to, for example, understand these options, right? When you want to, for example, with your eye, when you want to navigate through the gene and see where we have higher alignment and where we have alignment that is not of great quality, then you can do this. And if you remember earlier, we have not, for example, let's not click on this feature details. Let's see how, let's close this feature details. Okay, let's start enabling some, for example, some samples, 2 samples, and see what that actually gives us here. So when you do that, this is going to give us alignments, and it's going to help you visualize alignments in these specific regions. And now you can see that how the reads are aligned to this specific area, and let it load this alignment. And let's zoom out a little bit more. Okay, so you get more information here and the alignment. This is going to give you a plot or bar graph of the quality of the alignment that you will see. So you will discuss this in greater detail when we are going through that specific session. I just want to show you that you can qualitatively view this output and make sense of your this thing. Yeah, okay, we got the hill plot also. They're going to show you the coverage, and that's going to show you how much the alignment. Yeah, for example, we have more coverage in this area where we have peaks and less coverage in the area where we have troughs. So we will discuss a little bit in greater detail. And once you have also gotten your respective credentials, we would also use that in the session to do that also. Okay, let's quickly move on So that's about it. And you're also free to download this VCF file, right? I've already done that, and then open this in any text editor so that you can generally understand the structure of VCF file, how it is reported, and what are the other information, which will be very useful if you want to perform some downstream analysis in R and any other tool, right? Okay. With that, let's move on from genomics data analysis. And if you have any questions, this is regarding genomics data analysis, this is a time that you would want to ask me, and reserve your very specific and very detailed questions to the sessions. And here we are just having an overview, and I already gone over the time. Let's quickly go to the Bioinfo platform. So let's go back to the program page where once we have completed learning about genomics data analysis, then we are going to amp up the level of understanding to consider metagenomics data analysis, where we will be focusing on quantifying the microbial population using the metagenomics data. So how it is done and what are the considerations, all of this will be, for example, discussed in two sessions that we are focusing on metagenomics data analysis. So where to find respective demo pipeline for metagenomics excuse me, data analysis? You come all the way down in the metagenomics section. We have two analysis setup, one for KIME2 analysis, the other one for DADA2 analysis. So the difference between these will also be discussed in the program. So it will be a little bit out of scope for what we are dealing with. Okay. So let's take KIME2 results and okay, Malik, we are using the big data server, big data bioinformatics server that you will also use to perform this analysis, to set up pipelines of analysis in genomics, metagenomics, and transcriptomics data, right? When you get your access to this particular server after signing up in the Learn portal. So after signing up in the Learn portal, you will get the access after approximately or roughly 24 hours, and you can navigate the portal and access all the demo pipeline that I'm actually showing you. And when you are going to set up your own pipeline, and this is the reference that you will use to set up your own pipeline. Let's quickly go through and, for example, take let's take amplicon sequencing 16S 18S DADA2 analysis, and take analysis for samples with samples under chow dietary regime and samples under high fat dietary regime. So we start again. Now you would have gotten or understood the how the organization of these algorithms and into pipelines that we can use as click and select click and select selectable tools in this server is being used. So usually the analysis is actually separated and segregated into multiple sections. There is a color code to this section. The pre-processing and mapping and other colors will follow generic color code that is consistent in the whole bioinformatics, a big data bioinformatics server, T-Bioinfo server, right? Yes, Festus, we will actually help you build your own pipeline, right? If it is for your data, then we will help you build your own pipeline. And for the purpose of learning the different sections of the pipeline and what does it focus and what are the choices that one can make in setting up this analysis, right? So for those things, we will refer to the demo pipeline and yeah, and etc. And also there are multiple other pipelines, specific pipelines that are referred in the lesson while you are going through. So when you have your own data, then you can sign up to the research fellowship program, where you can perform analysis in with your own data and set up pipelines and communicate with me every week about what you have learned from your data and what is the focus of your research, etc. So yeah, for more information, please get in touch with us using support@pine.py. Okay, let's quickly go through the output. So when you set up such an analysis, right, metagenomics data analysis, what will you get? So that's a focus, right? How to navigate through these analysis, the different analysis option will be discussed in the respective sections. Let's speed up a little bit. So you have multiple visualization tool. So let's, for example, take one of these visualization tool, like relative abundance of taxa, and then external dashboard, which is also which is an interactive dashboard. Yes. So let it load for a while. Yes. So this is a typical abundance, proportionate abundance plot that you would have definitely noticed whenever you are going through a metagenomics paper or metagenomics research paper, right? Yeah, I will take these questions after this section. So in this one, you can navigate or you can understand the proportionate abundance of specific bacteria, and you can, for example, interactively find out what the bacterial genus is. This is like Bacteroidetes, right? Yes, Bacteroidetes phyla, and that is going to be abundant in some of my sample and not abundant in other samples. So I have to go to this sample and understand their sample condition to find their relevance. So that information can also be found from the dashboard. We have the same plot in a different facet here, and separated by the condition of a dietary condition that these samples are undergone. So I can find that, for example, one specific phyla, it's like this is Porphyrimona, right, which is abundant only in, or which is present only in the samples that take high fiber diet and is completely absent in the sample that takes chow diet. These are all the interesting implications that you can find and relations that you can find, that you can try to relate that with the diet. So you can also, for example, perform abundance analysis as a group, where you have you could, for example, select different conditions and find out how their distribution is and what their mean abundance level is, and what can we understand from this. We can generally find that the abundance is median abundance is lower for the sample that take high fiber diet compared to high fat diet compared to normal diet. Okay. Yes, we can perform alpha and beta diversity analysis, and okay, it's getting really specific. We will discuss in a short while. Okay, let's go up again. We can actually navigate through, for example, diversity visualization using NMDS or PCOA, depending upon the choice that you have opted for while performing this analysis. And you can, if you drop down, you will find options to select annotation at a different level. If your data has annotations at more than one level, like diet at different week, then we can use those different options to color and to shape this to find relevant patterns. So that's what we will discuss in the metagenomics data analysis, and also when to use what algorithm, like when to use KIME or when to use data2, etc. So this dashboard will also give you a tabular output, which you can interactively find and interactively download, so that if you want to perform a downstream analysis using using R or other Python code that we have also referred to in the respective sections and respective courses, you can use these input to perform those analysis. And we have all of this information in the respective courses, and will also be discussed in detail during this session videos. Okay, let's take some of the questions before quickly going through setting up of transcriptomics data analysis, what transcriptomics data or RNA-seq data can help us understand in terms of omics data analysis. Let's take some of the questions. We say, okay, what is quality control filtering during the process? Vasudha, I mentioned that it's about filtering your reads based on the quality score that is associated with each and every base call during the process of sequencing itself. The sequencer machine will associate each and every position with their respective quality score. And to maximize the potential, or to maximize the interpretation, and to maximize the statistical relevance of your data, we can actually restrict the reads or restrict the input file to specific quality score, so that lower quality base calls, which is more often related with, or which is more often ambiguous and maybe noise, or maybe due to signal overlap, will not be considered and will not actually disturb our initial signal. So, or will not for example, lower the statistical relevance that we want to find out. Okay, let's see. What's the next question that we have? What's the difference between genomics and metagenomics data analysis? In short, genomics data analysis is for sample from one organism, usually the organism that you are trying to study. It can be a pathogen like SARS-CoV-2 virus. It can be human, like the one that we performed in the cancer comparison analysis. Metagenomics data analysis is for a whole community of bacteria or microbes, more specifically, that is present in the different where you are sampling from. If you are sampling fecal samples, then you are trying to understand metagenomics of microbial community present in your gut. You can sample from oral cavity, then you are going to sample metagenomics of your saliva, and etc. etc. You get the drift, right? There we are not focusing on the host, which is going to be us, but we are focusing on the microbes that are living inside and on us. That is beneficial, and very few of them will be detrimental. And the idea is to find out the balance and relate that balance with the different disease condition, defines metagenomics data analysis, etc. That's where it differs from genomics data analysis. And can we do alpha and beta diversity analysis? Yes, we can do. And one of the way in which we can understand this difference in abundance level of microbes to specific phenotypical condition or to specific disease association or to conditional association is to study alpha and beta diversity using different parameters. Okay. And what is the bacterial resolution to know the significance between experimental groups? Okay, we are going to stick our analysis to what has been to the resolution of the Silva and very well known databases. So the resolution is how far can you resolve in your in your abundance on proportional table, right? If I have information and if I have annotation all the way to species, strain level, then I have higher resolution. But for some of the bacteria, the genus, species, strain information is not classified, and they will fall under lower resolution data with uncharacterized genus, species, and etc. other ranks. Yes, you will get the recording, and thank you. I think we are done with the questions. Let's move on. So in the next set of sessions, we are going to introduce you to the wonderful world of transcriptomics data analysis. And to give you a perspective, RNA-seq data or transcriptomics data is the most frequently used omics data type analysis till date. And I think very soon metagenomics will overtake that, but at least now transcriptomics usage is actually pretty well maintained. So we are first going to show you how to perform these again. Use the pipeline to quantify the gene expression, which is the whole idea of transcriptomics data analysis, to find out at what expression quantity each and every gene in my sample is expressed at, right? Some of the gene may be expressed at a higher quantity, and some of them may be expressed at a very baseline quantity, right? What is that quantification, and what can we learn from this quantification done on different sample conditions? My sample conditions can simply be, for example, transcriptomics data studied for people under diabetic and the people that are healthy, or it can also be people cancer patients who are treated with specific drug and cancer patients who are not treated with particular drug. So those kind of associations and those kind of differences and implications that you can make using transcriptomics data. But it doesn't stop there, right? Let's take a look at how to set up this wonderful pipeline. Right, under transcriptomics section, you come to bulk RNA-seq, and then you have several demo pipelines. Let's take one of them, cell lines. And now you get the drift, right? Genomics and transcriptomics share many of the algorithms because basically they are the same. Transcriptomics is a subset of your genome, right? Only the expressed part. So we again use the same pre-processing technique like trim the reads for adapter sequences and for unclean sequences or clean sequences with lower quality score, as somebody asked me. And then we again use PCR clean to drop out some of the PCR duplicates, and we go all the way to mapping onto transcripts, right? Now I'm completely skipping all this, but you will get a lot of information on what is mapping onto the genome and what is mapping onto the junction, what is exon detection, what is isoform construction, and what moving on, what is mapping onto the transcripts, how they are different, when to choose what algorithm during the course, right? We will get that information. And this is also available in the transcriptomics course, right, which is one of the most bulkier course that we have in the Learn portal. So you are free to check that out also. So once we have mapped the reads onto their respective places in the genome, that like what we do, instead of looking for differences as we have done in the genomics data analysis, now we are going to look for quantification of genes based on number of reads that are mapped onto those specific genes. That is done using these expression level quantification algorithm, which are plenty going to help you. Okay, it's meta-transcriptomics, right, Peter Festus? I think it is meta-transcriptomics. It's not transcriptomics, metagenomics. It's actually meta-transcriptomics, which is also very unique. And yeah, we are not going to deal a lot with meta-transcriptomics. If I have time, I'll tell you why. Okay, let's end the pipeline, and then you will get, for example, a wonderfully interactive dashboard, which I encourage all of you to check this out. Okay. So when we stop at this level, expression level, right, instead of completely skipping the downstream analysis like differential gene expression analysis, right, and we get until this interactive report, right? Somebody asked me about the quality of the reads, and this is one way to assess the quality of the reads, to check the multi-QC report that is going to be outputted, or that's going to be coming out of your analysis. There are several sections of this multi-QC report, but I'm going to show you this is a mapping of the read scores, quality scores for each and every position in your reads. You see how good this is in the green section or better area, right? These 27 or 30 will form the filter based on which you can filter out some of the positions that are going to be ambiguous in our downstream analysis. That's what is going to that is what filtering out based on QS or quality control of FASTQ file would mean. So this is one way. And then again, you can, for example, navigate the gene expression. Right? This is going to take a long time to load. So while it is loading, let's go back again here. I want to show you. I want to quickly show you, while performing this transcriptomics data analysis, your downstream analysis can form part of this transcriptomics pipeline. So in that case, we will also include these differential gene expression analysis, where you can statistically identify genes that are expressed at a different rate in one type of sample condition compared to the other type of sample condition, other sample condition. So that is the whole aim of using the quantification or quantifying your gene expression, is to compare at what level or what quantity the genes are expressed between my two different sample conditions or multiple different sample conditions. Usually it is two. Why? Because yeah, if it's more than two, there are different ways to set up the analysis. But again, that we will discuss during the session, or it will take a long time. Okay. When you perform such a pipeline or such a pipeline, yes, you will get these kind of results, where you will you can open up the dashboard. Now we have volcano plot and also dashboard, right? We will, let's see if this is loaded or not. This will take a long time to load. This is just a gene expression, visualization of gene expression for different samples. Okay, let's take the external dashboard. Okay, so here you will get heat map of top 20 genes that are expressed for your sample conditions, right? We will know that which sample belongs to what condition, and then based on which we can understand, okay, my top 20 genes can easily segregate between or separate between the sample conditions here. But this data is not that great, because we find that not many of the genes are identified as statistically significant from here, right? So we can find out what is this gene. This is a EGFR, okay, growth factor, understood, because I think this is a cancer data, cell lines data, and this is another gene, so DPYSL. You can come down. So since we have found out only two of these genes to be statistically significant and they are downregulated, upregulated, we can mention those genes here, EGFR R, and then, and what is the other gene? DPYSL2, right? DPY, yeah, okay. When you do this, then you can understand, look at this, both of them have a almost similar expression level, and they are extremely expressed at a lower quantity for these sample and expressed at a higher quantity for the other sample, right? That is what we can identify from this one. Usually when you perform this analysis using a different data set, you will have plenty of genes that are identified to be statistically significant and upregulated and downregulated. So we will get examples of that during the course and etc. So this comparative analysis of gene expression is just one part of downstream analysis that you can perform on transcriptomics data. And then we are going to focus on, for example, how to extend your data, or how to apply advanced statistical analysis like data mining approaches, like machine learning tools, to your structured data that you can get from metagenomics, that you can get from genomics data, that you can get from transcriptomics data, is what is going to be covered in the last few sessions. And we are going to introduce you to the concept and to the application of single cell transcriptomics, which is a world by itself. And we will also discuss what is the one that is different between single cell and bulk cell, and how is that going to improve our knowledge in understanding the biology of the samples that you are trying to study. And finally, we will conclude on how to find, where to find these publicly available data, and how to take some of these publicly available data and formulate a valid hypothesis and formulate a valid question for which you can design methodologies and then address those methodology in a research type internship, research internship program, which we will discuss a lot in this session. And also you can also get information about the research fellowship program from our Learn portal also, right? So I already took a lot of time and borrowed a lot of time from Ojasvi, and let me pass the stage to Ojasvi now. And if you have any questions, please put that up in the chat. So when we have time, we will discuss them after Ojasvi's session, or I will keep answering, or I will keep responding to your queries in the chat. Thank you very much for the interactive session, and let me pass the stage to Ojasvi, I think, right?
Yes, sir. So for now, to share details about the program registration and the scholarship form, I'd like to pass on the stage to Shubham. So the stage is all yours.
Thank you, Shrigari. So hello everyone. I am Shubham Kumar. I am the business development and the marketing associate here at Biotech. So let me first share my screen first. Yeah, Shrigari, could you confirm is my screen is visible?
We can see your Zoom screen. Can you share the program page?
Yeah. Is it visible now?
Not yet. It's showing Zoom.
Just a second. Yeah, now is it visible?
We can see your Zoom screen. Can you go to the program page and let me see if I can see that? Are you sharing the program page?
Yeah, I'm sharing the program page.
Okay, I'm not able to see that yet.
Just a second.
Yes, now it is loading up.
Okay. So hello everyone. So as you can see, this is a program page for Introduction to Bioinformatics, Multi-Omics. So I'll share you the link first. So you can go through this link after the session, and yes. So yes, as you can see on my screen, this is the program page for Introduction to Bioinformatics, Multi-Omics. Here you will get to learn about genomics, metagenomics, transcriptomics, and the machine learning also. So this it will commence from 23rd Jan till 30th April. So only 10 days are left. So I would request you to hurry up and enroll yourself. So after scrolling down a bit, you will see the program overview, where we have summarized what you will be learning with this program, along with the outcomes, like you will be able to know the analysis of NGS data, which will be helpful in identifying the pathogenic, germline, and somatic DNA variants. You will also be able to measure the gene expression and detect the methylation patterns, as just now Dr. Raghavendran sir has told, using the T-Bioinfo server. So basically, this program is designed for everyone, including the students who don't have a background in bioinformatics and the biologist, so that they can learn about the NGS data analysis program processing. So while scrolling down more, you will see an interactive calendar with some of the dates are highlighted. Like you can see, 23rd Jan is highlighted. So on that particular day, we will be conducting the live sessions. And on the right hand side of that calendar, you will see the topics to be covered in that particular session. Like in the first session, we will cover the introduction with the faculty and the program introduction. Then we will have the demonstration of the Slack channel and various program pages. Then Omics Logic Learn courses, then T-Bio platform also. And on the next session, which will be on 26, you will be learning about the genomic variants and mutations. So here you can see all the dates of your session and the topics which we'll be covering. Next, coming down a bit more, you will see that we have three different tenures: 45 days, 60 days, and 90 days. So, first, I'll tell you about the common resources which are present in all these tenures. The common resources are online tutorials, you will be have the access, then project examples also, then hands-on coding session, then session recordings. By chance, if you miss any of the session, then we will provide you with the session recordings. You will also get the online mentor support and the technical support. And after the completion of these tenures, you will get the certificate of completion. For 60 days, what extra you will be getting is one-on-one mentor session and a chance to propose your research work. And if you can complete a research project, then you can even present in as a poster on various international symposiums also. And in the 90 days, apart from these outcomes, you will get the one-on-one mentor session and an ample amount of time to work on your research project work and get it published in a peer-reviewed journal. So these are the difference between the 45 days, 60 days, and 90 days tenure. Coming to the pricing part, so as you can see, the prices are here in INR. So for 45 days, it is 7,500. For 60 days, it is 9,999, and for 90 days, it is 14,999. Now, if someone is from another country, like from USA or any other country, so you can simply select your currency by clicking on the drop down menu here, like just say in USD. So in USD, you can see the amounts like $150, 300, and $450. And for also for African participants, the prices are in Naira also. So now if anyone wants to like enroll for this program, so what basically you have to do is, like, let's say I'm interested in the 90 days tenure. So I'll simply click on buy now button, and here you will see your checkout page. So the currently we are providing 50% scholarship on all the tenures. So the coupon code for 50% scholarship is multi 50. Everything is in caps. Multi 50. Yeah. So after the coupon code, click on apply, and yeah, you can see the coupon is applied successfully. So just fill up your basic user details and click on this checkbox, and after that select the Stripe method or PayPal method. Also, if you want to pay by an alternative mode, then we also have alternative to proceed with the payment part. So if you have any query regarding the registration part, please let us know. By that time, I'll share you the steps to enter the coupon code in the chat box.
Thank you so much, Shubham. So meanwhile, Shubham shares the steps for registering. If anyone has any queries, let me know. Malik is asking, what about group registration?
Yeah, we also have the group registration also. So Malik, could you please send me your email ID? We will revert back to you with the process for group registration part.
Yes, Malik has already shared his email ID, so we'll be getting back to you. That.
Okay. Yeah.
So does anyone have any further queries regarding registration or scholarship? Let us know in the chat and we'll be happy to help.
Can you please share the registration link one more time? I think, okay, you have shared. Thank you. I just found it. Yeah.
Yes. Yes.
For any queries, I'm sharing the email ID and the contact number of our marketing manager. So at any point of time, if you're having any queries, you can reach out to us through this email ID or contact number, and the number is also active on WhatsApp. So we'll pause for a couple of seconds for the participants to share their query. If not, we'll be ending today's webinar. Thank you, Malik.
I think, wait a second. I think Sharona has a question. Yes, Sharona, we will share the recordings under each and every session details, right? One thing that I forgot to mention is under the resources section of, let me share it quickly. I think, do I have it or have I closed it? I think it seems like a, has anybody gotten your program page, Srigauri or Ojasvi?
Yes, sir. Yeah.
Yeah. Can you
I'm
share it?
sharing the screen, sir. Yes, sir. Just a second. Yes, sir.
Thank you, Shubham. Scroll down. So for example, here under these associated resources, like, no, not there. Go to the go up, scroll up. Scroll up to the, go to the first session, please. Yes, okay. So here you see that there are the list of associated online resources. So every time when a session is completed, give it 24 hours, then you will get the session recording appearing below that associated online resources. You can go over the session recording at your own leisure if you have missed the online session, live session, okay? And some of the sessions are planned to be asynchronous, meaning you will get a link for the videos through email, or that link will also be updated on that specific date of the session has been planned to under these online associated online resources. If you register to this program, you will get an option to click and select complete each and every session, so that we also can keep a tab on how far are you progressing with the program and etc. So once you register for the program, you will find these two options under these under each and every one of the session. Thank you, Shubham.
Thank you, sir.
Thank you, sir. All right. And if anyone has any further queries, let us know. I'd be happy to help. All right. If the participants have no further queries, ending today's webinar. So thank you everyone for joining today's webinar, and we look forward to seeing you at program start. All right. Thank you everyone. Have a great day.